Good morning, everyone. It's dot 10:30 a.m. and we would like to start this National Symposium on Retina. And the topic of National Symposium is none else than anti-VEGF drugs, which is a hot topic for all the ophthalmologists, irrespective of the specialty they practice. Because not only retina people, but all the comprehensive ophthalmologists also uh, give these injections. So hence the importance of this symposium, the use and abuse of anti-VEGF drugs. You see, we know that various trials are available which justify their usage in ophthalmology, and this has been around for more than 1.5 decades. An entrance of biosimilars not only in India but abroad. All of you know that USA recently approved two ranibizumab biosimilars, have made this treatment very competitive and may I say lucrative also, because there's a lot of finances involved in this. Further, newer anti-VEGFs are in the pipeline, which give us hope to reduce the treatment burden. And remember, all these injections are of a temporary nature. None of them has a lasting effect for life. They don't cure the disease. They just alter the edema or the angiogenic factor. And financial aspect, believe me, have increased the abuse also. Irrespective of indication, whatever you see in the macula, retina, people are somehow fond of giving this injection. जैसे just एक लगा के देख लेते हैं, which is not good actually, not scientific. So through this national symposium, we'll try to rationalize the usage for the maximal benefit of our patients. With this brief intro, we'll get going with Dr. Santosh. And I'm very fortunate to have on my left Dr. Mallika Goyal, needs no introduction, and Dr. Joshi on my right, who will help me in conduction of this national symposium. So, Gopi, if you're ready, we'll
Yes, sir.
get
Yes.
going.
Good morning, all. At the outset, I would like to thank Lalit sir for this opportunity. It's an honor to be presenting here. So I'll be talking about an update on an overview about angiogenesis. I'll try not to indulge too much into the basics of it. So the basic definition of angiogenesis is formation of new blood vessels from pre-existing ones. It involves migration, growth, and differentiation of endothelial cells. As you see here, it just starts, and this is how the blood vessels actually start growing. So it's not just pathological, it's even physiological. It occurs throughout the life in both health and disease. So that's the main thing that we need to understand about angiogenesis. When we talk about angiogenesis, the other thing that comes into mind is vasculogenesis. Vasculogenesis is a normal physiological thing which happens to denote development of blood vessels, especially in the development of primitive heart and major blood vessels. And it starts from at the first and second trimester. So once a new blood vessels start forming, from there angiogenesis takes over. So this is how it happens in 6th week and 18th week. You can see excessive branching angiogenesis, and this is a more stabilized angiogenesis at term pregnancy at the bottom figure. So angiogenesis, again, there are different types. Sprouting angiogenesis was discovered some 200 years back. This is how it sprouts. So when you have an angiogenic stimulus, the hypoxia, there is a breach in the blood vessels, and endothelial cells start growing. So there is another concept called intussusceptive angiogenesis, wherein the blood vessels either split and or they kind of split in between, and then they start growing. So vascular co-option is another phenomenon, especially when we talk about angiogenesis. Most of it is regarding tumors. So this is how the tumor cells grow around the blood vessels, and then they start differentiating. Vascular mimicry, I'll come to detail in another slide. So the basic differences between physiological and pathological angiogenesis. Physiological is because of metabolic demand, shear stress, and hormones. So both physiological and pathological result in increase in blood vessels, wherein you can see a normal branching pattern and normal network pattern length in the physiological thing, whereas you can see a disturbed and abnormal branching as well as abnormal length pattern over here. So therapeutic aspects, when you look at angiogenesis, it's extremely important. Where all do they happen? Physiologically, it's important to have physiological angiogenesis, that is a healthy angiogenesis in wound healing in patients with myocardial ischemia or peripheral artery disease. And in cases like peripheral artery disease, we usually there is something called therapeutic angiogenesis, wherein there is a angiogenesis is induced. So angiogenesis is preferably inhibited. I mean, the preferable choice would be in cases like tumors, cancers, or when it comes to retina, DME, and even AMD, and even in rheumatoid arthritis. So this is a complex interplay of the factors what you see here. So how it starts, VEGF, MMP, and initially the vessel kind of dilates, and then it goes in for proliferation and migration, and you can see the interplay of complex factors, and these are the factors that are anti-angiogenic, which can stop these things at different phases. Over here, they see migration and proliferation and tube formation. So this is a cascade of events. The initial stimulus, and slowly the endothelium starts growing, and you can see the cells growing towards the angiogenic stimulus, the hypoxia or tumor cells. And when you look at normal wound healing and an impaired wound healing, especially in cases like diabetes, burn injury, or ischemia, you see that the angiogenic cyst kind of comes down in impaired wound healing, and the inflammatory infiltrate kind of goes up, and there's a impaired collagen synthesis which results in impaired wound healing. So there is again a risk of relapse and recurrences when you look into these angiogenesis processes, even when you kind of try to balance these things. So angiogenesis usually happens when you have the pro-angiogenic factors which are more than the anti-angiogenic. So that's the reason you get these abnormal blood vessels. And when you kind of treat these things with an anti-angiogenic drug, there is always a chance the balance can shift and it can come back to again abnormal blood vessels. So this is a balancing act. It's a very fine balancing act. As you can see, the initial stimulus being hypoxia, and this is how it usually progresses. So many factors that are involved, and these are the inhibitors. So it's a fine balance that is maintained in the normal healthy person. So angiogenic switch is wherein you have this angiogenic factors more than anti-angiogenic factors, and that is how the blood vessels start growing, and tumor cells kind of fill. When the switch is off, then the balance is maintained, but the switch is on, then again angiogenesis starts. So this is vasculogenic mimicry, what I was talking about in tumor cells, wherein the tumor cells themselves behave like a vessel-like channels. So these are the key milestones in angiogenesis research and drug discovery. It started way back in 1945, and then 1948, wherein there was a hypothesis of something causing diabetic retinopathy. Angiogenin, 1989, isolation of VEGF. Then it went on to the initial pegaptenib, then again ranibizumab, and then the story follows. So we all know what happens in these conditions, which is important for retina surgeons because of these issues that are strongly involved in DME and PDR, as well as in AMD. It's a complex issue, interplay of things. So it's difficult to understand, but if you know the basics of it, it really helps. So the available options are these. The other speakers, following speakers, will go into much detail. So these are the current anti-VEGF agents, and these are the retinal diseases molecular signatures. So if you look at neovascular AMD, DME, PDR, and RVO, these are the molecular signatures, and different angiogenic and pro-angiogenic and anti-angiogenic factors play a great role in such conditions. So looking at multiple targets in angiogenesis, so at every step you have different factors that could be targeted to prevent or kind of stop this angiogenesis. So this is how the hypoxia and proteolytic degradation and kind of forms a sprout. So these can be touched. Some are explored, some we do have drugs, some are still under research. So these are the potential targets. So if you look at VEGF receptors and tyrosine kinase and FGF, and so these are all the ones which are already kind of the drugs had come out, and these are still yet to go through the approval stages. So these are the treatment options and the drugs in pipeline and different phases. So I'll just, before finishing, I'll just go through quick case examples wherein we had kind of looked at these factors and try to implement in the clinical practice. So it's a incomplete resolution of a CME. It's a persisting CME, and when you look at the array of molecular factors, what we usually do at our place, you can see the VEGF to be on the higher side, and the basic ones like IL-8 and the ICAMs and VCAMs are also on the higher side. Since the inflammatory factors were more, the IVT was given, and you could see the resolution. So, but when we took the patient had come in a year later, you can see that the VEGF had come down. So the factors now have changed. The addition molecules were on the higher side. So this is a comparison across first visit and second visit. It's a consecutive thing. You can see the variations in these levels. So when you kind of get to know that what really causes the macular edema, sometimes recurring edema, especially these things can result in better addressing these issues and targeted therapy if possible. Then again, second same case, glaucoma issues, patient uncontrolled diabetes and recurring CME. So you can see the VEGF to be on the higher side initially, and all other factors also on the higher side. So with glaucoma medications, we went ahead and gave an IVTA. So this is how it was. So you can see the VEGF to be completely down initially, but other factors were on the higher side. So third analysis again was done post second injection. So you can see the factors to be VEGF was almost nil, whereas the ICAMs also, the addition molecule also came down over a period of time. So then again, these kind of practical things suggest that there could be future possibilities of wherein we can target these things and get a better treatment outcome. So to conclude, there are different therapies which can be looked into. You can look into pro and antigenic factors and cell therapies and microRNAs and attractive and repulsive vascular guided cues. So it's a complex process. It's highly relevant in routine retinal pathology. So most probably multiple targets, and maybe 10 years from now, maybe we'll be looking at varied agents and newer guidelines to treat these conditions better. Thank you so much. Thanks a lot.
Thanks, Gopi, for covering this in 10 minutes, what books have been written on vasculogenesis. You see, the gist, whatever he has told us, is that angiogenesis is required for normal, healthy living also. It is when it occurs in a pathology situation like retina, then we get bothered. And the stimulus is hypoxia or inflammation. But unlike healthy angiogenesis, what term he used, these unhealthy vessels or pathological vessels, they are not like God-given vessels. So these vessels are leaky, these vessels are fragile. Leaky means you can have fluid outside, and fragile means they can, rupture. And all attempts are being made to, have control over all these things, and hence the role of anti-VEGFs came up. It all started a couple of decades back. We have had so many anti-VEGFs now available, and there is a very strong tug of war going on between various anti-VEGF agents available. Any question, Malika, you have? Yeah.
Very nice talk, Dr. Santosh. Since you are doing a lot of research on the angiogenesis and you have a grow lab at your institute, so you nicely showed what are the pro-inflammatory mediators. It could be interleukins, MCPs, ICAM, TNF, alpha, or many other factors. So is there any way that you can find out that, okay, these are more of the pro-inflammatory kind of disease in this patient? And how strong are these anti-VEGFs? Because more basically they are more of VEGF mediated and neutralizing the VEGF receptors.
Good.
So apart from the OCT biomarkers, is there any other way to tell that, okay, in these patients the steroids works better, or in these patients the anti-VEGF works better?
Now, if you look at OCT biomarkers again, I mean, we did have a small pilot study wherein we looked at the types of edema and what are these factors that were elevated, but it was a small study. So the only thing is, as Sir was also saying, it's a complex interplay. So it's really difficult to kind of point out and say that, okay, this is a factor that's really causing this. If the patient had already received an anti-VEGF injection, obviously the VEGF load comes down, whereas other factors might be elevated. IL-8 and IL-6 are known to be associated with chronic
I think we have a topic for Tirumalesh, what has
right,
been
sir.
started.
Yeah, I think he'll be talking about those also.
Can we, road ahead, can we titrate, prognosticate based on
Correct.
whatever research you people have done regarding targeted therapy or which which cytokines, and can we inhibit specifically and which disease they are elevated. So thanks, Gopi, and
Thank you, sir.
Thank
we
you very
will
much.
move on.
Thank you.
And, uh, and after having learnt about angiogenesis and VEGF or other cytokines, we will ask Anand to tell us the use of anti-VEGFs in a broad way in various retinal diseases. I know all of you are aware about, 3, 4 common conditions, including ROP, where, anti-VEGFs have been approved, starting from diabetic retinopathy, diabetic macular edema. Even the process of diabetic retinopathy per se, they think that they can reverse. And myopic CNVMs and, so many other conditions. So Dr. Anand will give us a broad overview of use of anti-VEGF in various retinal conditions.
Thank you, Dr. Lalit. It's an absolute pleasure and honor to be talking to all of you today about the use of anti-VEGF agents in retinal diseases. Thank you, Dr. Lalit, sir, for having me here. So, for centuries, mankind has been in pursuit of this elusive holy grail. But us retina specialists have been particularly fortunate in finding our holy grail in this anti-VEGF agents for retinal diseases. And many of us have kind of called it the Brahmastra for retinal pathologies. Some have likened it to that God particle, which kind of justifies and rationalizes our existence as retinal physicians. So, anti-VEGF agents have had an unprecedented and incredible impact in the reduction of global blindness. So, observational studies have shown that across various European countries, almost there's been a 50% reduction in the incidence of blindness over the last decade. That's a tremendous amount. Theoretical modeling studies have shown boosted that number to up to almost 72% in the US. So these are the key agents we're fairly familiar: ranibizumab, Accentrix, Lucentis, bevacizumab, or Avastin, aflibercept, or Eylea. And this is how we're used to seeing it packaged. Their key anti-VEGF agent structures, well, all of them target VEGF-A isoforms, but aflibercept additionally acts against VEGF-B as well as placental growth factor. Ranibizumab is a FAB fragment, bevacizumab is a full-length monoclonal antibody, and aflibercept additionally is a fusion protein. And these are the doses we use them in. So cutting to the chase, moving to DME, the management of DME is moved from the erstwhile laser photocoagulation in the 80s through the turn of the millennium, where intravitreal steroids, namely triamcinolone, ushered in the pharmacotherapy era, to this current era of anti-VEGF agents. So for DME management, guiding principles from the trials, what works? We know based on these large, big trials that anti-VEGFs have high efficacy versus sham, especially ranibizumab and aflibercept. But what works best? Which one do we choose? So DRCR protocol T gave us two important statistical highlights. A, higher efficacy for aflibercept and ranibizumab, and in this, aflibercept had a marginally better efficacy than ranibizumab over bevacizumab in patients with acuity less than or equal to 20/50 or 612 parts. Corollary is that in patients with better than 612 parts, you could use either. But bevacizumab reduces of the three macular thickness the least. Now, what works if the patient presents late? And this is important. If the patient comes with chronic edema or with the chronic previously lasered diabetic macular edema, what do we do? So this is a case which kind of highlights that a patient came in with poor control, couldn't take injections immediately, came in with the worsening. But then, after we persuaded the patient over 8 months, took 6 injections, ranibizumab, you can see a beautiful reduction. So this tells us that visual acuity does improve in untreated chronic macular edema with anti-VEGF treatment. And this is also seen in literature. In the RISE and RIDE trials, where you have these two ranibizumabs shooting off as soon as straight away from the start, and the no treatment are hardly getting off that baseline up to 2 years. And the moment it was open to ranibizumab treatment, you see that snaking up. And the same thing was seen with macular edema reduction. The moment ranibizumab is offered, you see that those maculae thinning out. Now, chronic lasered diabetic macular edema, another case which wasn't without treatment. But then, moment the patient was allowed anti-VEGF injections or accepted and took that religiously, 5 injections, 4 ranibizumab, 1 bevacizumab, you see that macular edema resolved beautifully, telling us that visual acuity will improve in lasered chronic macular edema with persistent anti-VEGF therapy. And this is also finds a parallel in literature in the Restore extension study, where you again see the two ranibizumab arms. The yellow one is the ranibizumab only arm, red is the ranibizumab combination laser at the start, and the blue is the laser only arm, which barely just hugs the baseline. And again, after 12 months, when ranibizumab is introduced, you see that line snaking up and catching up with the ranibizumab combination arm at the end of 3 years.
So this tells us that chronicity is not big issue with lasered edemas. Also, second corollary, that it doesn't seem to make a difference whether you put that ranibizumab at the start or 1 year later. At the end of 3 years, you seem to be able to make cut up. So in untreated eyes as well as laser-only eyes, it's never too late for anti-VEGF therapy. Also, interestingly, Restore showed us that adding laser didn't seem to reduce the ranibizumab retreatment rates, which we think is a USP. What else works? What about lasers and steroids? So DRCR protocol I showed us that the laser ranibizumab combination was better than laser or IVTA laser, and deferring combination laser was better than prompt laser. It also showed us that early intense treatment, getting a hold of this beast early on with intense injection, reduces the number of treatment burden later in the ensuing years. There are obviously higher safety issues for IVTA in terms of cataractogenesis and IOP elevation. The 3 regimens, monthly, PRN, TREX. How do we go? So with monthly dosing, we know it's the most efficacious, but it's obviously impractical and cost ineffective. Now, comparing treat and extend, this was done with PRN in the retained study, and a long story short, treat and extend was comparable to PRN regimen for DME treatment. That's fine if the dosing is inadequate. But what if there's adequate dosing and the patient is still refractory? Then you can go in for switching the drugs, Avastin to Axentrix or Eylea. There's an example of a case refractory to Avastin which benefited with Axentrix. Converse is also true, but not very common. But of late, a lot of series have shown us that aflibercept has great value in patients with DME refractory to Avastin and Axentrix. Now, moving to PDR, the debate, PRP versus anti-VEGF. So protocol has showed us that ranibizumab had non-inferiority to PRP, and that the ranibizumab arms actually did better in terms of acuity change from baseline, lesser peripheral visual field loss, fewer vitrectomies, lesser incidence of DME development. But let's just put it out here that in the Indian scenario, in a resource-challenged country like India, PRP is still the gold standard for PDR. And other uses also for refractory NV post PRP, you can put in anti-VEGF. Florid NV also, but please, a word of caution, this can sometimes lead to tractional detachments or crunch, so you have to be careful here. Anti-VEGF in florid NV in macular TRDs as a preoperative adjuvant, we usually do it 3 days pre-surgery. So this massive neovascularization, you can regress it, and then you can have a clean surgery post. Florid NVI also, anti-VEGF can wipe out or regress that neovascularization, as you can see a beautiful example here. And this is useful, especially in cases where you're going in for a trab, for neovascular glaucoma. So giving a pre-op injection will help. Moving to ARMD, evolution of standards of care for neovascular AMD have again moved from laser photocoagulation in the 80s through the turn of the millennium, where we had PDT holding sway, to the current 1.5 decades of anti-VEGF era. So the treatment regimens have moved from monthly through, we dabbled with quarterly, bi-monthly, and then PRN held sway for a long time. And the last 4 or 5 years, treat and extend has come to the fore. So I'm going to focus on the CAT study, which told us a lot. This was a government-sponsored study, NEI-sponsored, not a pharma company trial. And basically, you had 1 plus PRN versus monthly at the start. Now, at the end of 1 year, what happened was the monthly arm was broken into continued monthly as well as a switch to PRN. So you essentially had 3 arms for both the drugs, ranibizumab, bevacizumab, continued monthly, continued PRN from the start, 1 plus PRN, and then 12 months and PRN. Now, how are the results like? So at the end of 2 years, you see ranibizumab monthly had a fractionally better advantage over bevacizumab monthly. But interestingly, if you look at where the green arrows are, if the study had stopped there, then actually bevacizumab monthly was better than ranibizumab monthly. Now, this is a very important slide which shows you that in terms of equity, 1 plus PRN is equivalent to 12 months plus PRN. You see this for both the drugs, bevacizumab and ranibizumab. Look at the green halos. Look at the drug endpoint. Both reach the same point. So this 3 plus PRN doesn't really come into the picture when 1 plus PRN is equivalent to 12 months loading dose plus PRN. So the concept of loading dose was actually negated by government-approved study CAT, but still the label continues with 3. And the same thing was seen with macular edema reduction too, especially for ranibizumab. You see the green halo. In terms of OCT results, no fluid was seen on OCT in up to 45.5% in Lucentis monthly group. But what does this also mean? The corollary is that more than 54% had fluid on OCT. So the most intense treatment regimen will still have residual fluid in over half the cases. So if you're seeing fluid still in an arm which in patients whom you've treated very adequately, don't get discouraged. This will happen. You don't essentially have to get every macula bone dry. Also, what is the corollary there? What's the other flip side? Greatest proportion of study eyes with new geographic atrophy was seen in Lucentis monthly rather than the other lesser arms. So greatest among patients treated monthly. So this most intense treatment regimen raised the incidence of geographic atrophy, raising the question of whether a stronger drug given very frequently can cause some level of apoptosis. The CAT 5-year follow-up. Now, when the patients went off this very intense scrutiny study, you actually found patients going worse. Mean acuity declined to 11 letters worse than at 2 years, 20% acuity, 2200 or worse. Mean lesion size increased by 4.8 millimeters square from year 2 to year 5. FFA leakage in a quarter of those patients, geographic atrophy 41%, and no real difference between the drug dosing regimen. So real world experience tails off, and we'll see this later also. Coming to aflibercept studies, view 1 and view 2 pitted ranibizumab monthly, 0.5 milligrams against 3 arms of aflibercept, 2 milligrams and 0.5 milligrams monthly, and 2Q for 8 weeks. So they found that all the arms did equally well, as you can see. Key results being aflibercept 2Q 8 weekly demonstrated similar efficacy and safety to the standard then 0.5 milligrams monthly. But interestingly, 48% of patients in aflibercept 2Q8 could be maintained on a quarterly dosing. And during the second year, fewer patients in the aflibercept 2Q8 group received 6 or more injections compared to ranibizumab. So we've been talking about safety, efficacy, cost, but now aflibercept was introduced for the first time a newer concept, durability. So that's been its USP. Now looking at treat and extend regimens like I mentioned earlier. So trend showed us that comparative anatomical outcomes can be expected with ranibizumab treat and extend and monthly. CanTreat showed that along with comparative activity gains, you'd get fewer injections. A bunch of studies showed that visual gains will be the same with treat and extend compared to PRN. And this very nice study from Japan showed that treat and extend very clearly reduced hospital visits compared with PRN and is associated with reduction trend in caregiver burden, including time and cost. So you see the blue boxes of TNE are much smaller than the gray ones of BRN. So basically TNE was cost efficacious compared to monthly and PRN. Choice of drug, rival showed that ranibizumab was equivalent to aflibercept in terms of acuity gain, macular edema reduction, re-injections, geographic atrophy, as well as safety. Altair also reflected somewhat similar results, but showed up the durability of aflibercept with up to 60% being maintained on 12-week intervals and up to 46% on 16-week intervals. And this slide clearly shows that it's so difficult to replicate what is seen in randomized clinical trials. In real-world evidence, the equity gains really tail off. So treatment choice, obviously in primary treatment, patient can afford it, then ranibizumab or aflibercept, perhaps aflibercept for durability. Cannot afford Avastin for refractory cases and large PEDs, aflibercept. Now, moving to PCV, the Everest showed us clearly from 6-month studies and the 2-year study that combination ranibizumab and PDT was superior to ranibizumab monotherapy in equity improvement at 24 months, as well as achieving complete polyp regression. The PLANET study showed us the 3 monthly doses of aflibercept followed by PDT or no PDT in the 2 arms showed impressive equity gains of greater than 10 letters maintained over 2 years. And more than 30% of patients receiving aflibercept monotherapy had complete polyp regression at week 52 and 96, and over 80% showed no evidence of active polyps at these 2 time points, something very important for us physicians. So aflibercept, as we've all seen, has a wow effect. It's a formidable flattener of PEDs, as you can see here. And several trials across the board, across the world, have shown impressive equity gains, as well as complete polyp regression. Ranibizumab, to maintain the balance, also has shown impressive results, especially the laptop study from Japan. And in summary, repeated anti-VEGF therapy can regress polyps, approximately a quarter of them completely, and 40%, 42% partly. And aflibercept, based on PLANET, shows remarkable efficacy and has pole position today for PCV management because we don't have PDT. Ranibizumab is also efficacious because of the laptop study. Submacular hemorrhage, you can use pneumatic displacement, tissue plasminogen activity in combination with anti-VEGF. And re-emergence of angio-occlusive therapy like PDT in combination is dearly awaited. In the meantime, low-cost alternatives like TTT or laser can be used, especially when the lesions are extramacular. Moving to retinal vascular occlusions, Bravo and Cruz showed the efficacy of ranibizumab for BRVOs and CRVOs. These two studies tailed into the Horizon, which was an extension, and then Retain, which is a further extension of this. So suffice it to just talk about Retain, which showed impressive results for ranibizumab in BRVO and CRVO at 4 years. Shore showed flexible dosing of ranibizumab. PRN is useful, only minimal injection. Now the question of effect of ischemia and disease duration or chronicity on anti-VEGF in RVOs. So Brighter and Crystal showed very clearly that retinal ischemia did not affect equity gains in ranibizumab treatments with BRVO and CRVO, and also that all these gains, impressive gains, were possible irrespective of disease duration. So basically, chronicity and ischemia should not deter you from trying out anti-VEGF for retinal vascular occlusions. So the LEVO study answered a $1 million question, the shootout between the big three. So it showed that aflibercept achieved numerically greater equity gains with significantly fewer injections than ranibizumab or bevacizumab over 100 weeks in CRVO patients. And Newton showed that aflibercept TNE in CRVO worked well for macular edema refractory to ranibizumab or bevacizumab. And following the switch to aflibercept, the macular edema free interval significantly increased in patients with CRVO up to a mean of 64 days. So the key takeaways in RVO: initial intense anti-VEGF therapy, 3 months followed by PRN is the way. Aflibercept and ranibizumab, more than bevacizumab, are effective as potent primary therapies in vascular occlusions. Ischemia and chronicity is not a contraindication for anti-VEGF therapy. Aflibercept have shown efficacy in RVO macular edema resistant or refractory to ranibizumab, bevacizumab. Dexamethasone and insulin may be considered as alternatives. Dexamethasone plus PRN anti-VEGF has shown to have faster and sustained resolution of macular edema. Steroid and induced IOPRI cataract is obviously an issue and is indicated in kind of elderly and pseudophakics. Can be used in relative contraindications of anti-VEGF, where, in example, relative stroke, acute MI, uncontrolled. So I'll take a minute. So moving to our last indication, ROP, it's a global problem. In India, over 2 lakh children are at risk of developing ROP every year. The anti-VEGF studies beat ROP, CARE ROP, Rainbow being the main ones, beat ROP, compared bevacizumab 0.625 with laser therapy. CARE ROP was a smaller study in Germany, but importantly looked at 2 doses in bilateral AP ROP, and ranibizumab looked at a large study versus 2 doses, 0.2 and 0.1 versus laser. So the beat showed that bevacizumab was safe. Bevacizumab monotherapy versus lasers in infants with stage 3 plus ROP showed a significant benefit for zone 1, but not zone 2 disease. Ranibizumab's conclusion in the Rainbow, treatment success rate of 80% was achieved with ranibizumab 0.2 versus 66% with laser. Infants treated with ranibizumab 0.2 were twice as likely to achieve treatment success versus laser, which is considered clinically relevant. Ranibizumab 0.2 and 0.1 were well tolerated in patients with ROP, and safety profile was as expected in a preterm population. We're looking forward to the 5-year extension results. Firefly is a study which was done for aflibercept in ROP, and the results were not great. Aflibercept were not non-inferior to laser, but surprisingly, aflibercept has been approved for ROP in Japan. Butterfly is a phase 3 ongoing study. The Cochrane review.
Can we wind up, Doctor?
Yeah, just a few slides. Anti-VEGF shows that there's reduced risk of refractive errors, significantly reduces risk of recurrence zone 1 ROP, but there is increased risk of recurrence in zone 2 ROP. So in ROP, finally, the key takeaways are: gold standard in the management is still laser photocoagulation, but has its own limitations of myopia, peripheral field restriction, macular dragging, hazy media. Anti-VEGF may be used as primary therapy in AP ROP and posterior severe ROP. Adjuvant role for laser is there when you have residual peripheral avascularity persisting, non-compliance for follow-up, or suboptimal response with anti-VEGF. So in conclusion, anti-VEGF has proved to be immensely efficacious in the treatment of vast variety of retinal neovascular and edematous pathologies, but importantly, warrants judicious use based on accepted clinical evidence, and is therefore it's become an indispensable part of our armamentarium in treating millions of patients globally. Thank you.
Thank you, Anand, for this wonderful. I just wish that we had more time. And as Ashish Sharma gets ready, only 1 or 2 compact points that Holy Grail, definitely yes. There is a definite impact in reduction of global blindness. But remember, whatever has been told is based on all this RCTs, randomized controlled data, or what I call red carpet treatment, because these patients, on trials are called every time. So therefore, instead of RCT, randomized control trials, we call them red carpet kind of treatment, which they get. But all of us are aware of the Lumina study. The real world scenario is totally different than this. And you see, a lot of information is there on this. But the biosimilars entered primarily to improve the compliance. You see, key issue is compliance. Key issue is finance, ultimately. So I'll request Ashish to please come in and tell us today, biosimilars, what is the role? Because affordability is an issue, is a very, very big issue. Yes, Ashish. And Ashish, we know, has done a lot of work. He is one-man authority on biosimilars in this country.
So good morning, ladies and gentlemen, and thank you, Dr. Lalit, for giving me this opportunity to be a part of this wonderful meeting. So we have been trying to understand biosimilars since last 4 to 5 years closely, and I would be going to talk about not only Indian biosimilars, but I would also be touching the what is happening overall across the globe as far as biosimilars are concerned. How do I advance this? Okay. So this is our contribution in the form of various publications. We have been trying to understand biosimilars from different point of view, whether it's an endotoxin, whether it's inflammation, what is a phase 3 trial design, what is the kind of awareness that you have not only in India, but across the globe. So this is what that we could contribute, and I would definitely like to acknowledge this fantastic group of people across the globe who have helped us in understanding these molecules, because they were in development stage when this country was already using them. So before I start, I would like to have a small kind of poll here, that how many of you, if I can have raise of hands, are user of biosimilar ranibizumab, which is available in this country? Brilliant. So at least I see 70% of audience is using biosimilar ranibizumab. I'll go further one step. How many of you use Razumab, which is from Intas? How many of you use Reniase, which is from Lupin? How many of you use another molecule from Reliance, Renizorail? So this shows here Intas is leading. Obviously, they were first, and we are proud that that was a company which has established this country as a leader in biosimilar world in 2015-16. That's why you see 70% of users. I was in a different session with Dr. Dogra yesterday, and once he showed the statistics of PGI Chandigarh, they had highest usage of biosimilars, then comes the innovator. So that's what biosimilars have done in this country. And you see Renia is coming on second, and Renizorail comes third. So biosimilar overall, just for sake of saying, but as of now, we know that this country understands biosimilars better than anywhere in the world. So these are the similar molecules, but they are not exactly the same molecule. But the problem lies whenever you talk about that these are not similar molecules, that's where the negative perception comes. But if you really see, if a molecule, if I say, Lucentis, if I get it from Europe, if I get it from US, they would be different. Because there is a different machinery. These go through the live cells. So two batches cannot be same. But regulatory requires that you need to prove that these differences are not clinically meaningful. Clinically meaningful means they do not differ in terms of clinical efficacy and safety. Why biosimilars? Dr. Rajendran has put up nice presentation of all the innovator molecules just now. But can we afford them? Can we afford the kind of regime? As Dr. Lalit rightly said, real world is different. Our average injection rate is approximately 3. But what biosimilars have done, because of the cost benefit, you can inject many more. I can treat my father with confidence with continuous injections now, which I was not before. Now, what is the status? As of now, across the globe, only biosimilar ranibizumab is available because the patent is expired. But aflibercept biosimilars would be available in 2027. That is going to make a big change across the globe, especially to this country. Entire world uses Eylea at this point as a predominant user. We do not use that, again, because of the price point, in spite of having a great drug. But we would change, the dynamics would completely change once biosimilar aflibercept are available. As of now, just for understanding, there are 3 companies which make biosimilars: Resumab, Byintas, Renia by Lupin. You must have heard the name Osiva. Somebody is coming to your door. That is the same product because Lupin manufactures it. As of now, Sun Pharma distributes it. Ranizurel from Reliance Life Sciences, they make it. Cipla distribute it. Japan, you have another biosimilar named Ranibizumab BS1. US and UK, everybody was waiting for this because people were having negative perception about India. But once it started, since 2 years, 2 drugs are approved. SB11 BioViz, same name by US FDA and EMA. Another molecule which was known as FYB201 in trial, but they have different name in different countries. For example, I was in a different session, Dr. Jaishankar was there. So now NHS has designed a step therapy that biosimilar ranibizumab has to be used first. And it is known as Ongavia because everywhere you need to preserve finance. That can be allotted in something different. You can increase manpower. In US FDA, it is Cimerelli. Ranibizio in EMA. So they are different name, but the same drug, just for the understanding. Is there a differentiator for clinicians? Means sometimes you have a confusion that what should I use? If you look at the clinical trial design in India, last 3 months, I would as of now say that Renia had the best clinical trial design till now. They got award yesterday. Because it was apple to apple, same kind of, patients, number of patients in both the arms. But we have grown up. It's not, the previous trials were not great, but because we have evolved, understanding has evolved. Now you look at the US trials, they had more number of patients. And they do trial till 12 months. Indian trials were till 3 months, 6 months. But does that make Indian molecules inferior? No, it all depends on what your regulatory wants and what this country needs. This country needed things, approval faster. Now next question comes, do we have real world data? As of now, molecules which are approved by FDA, EMA, we do not have real world data. Japanese molecule, we do not have real world data. I am in close relationship with them. Probably we would bring it. Indian biosimilars, yes. Ranizurel, we published this with their limited data. You cannot really establish safety, but I would touch on the safety part as well. Renia started well. Now they have almost 15,000 injections without any safety issues. Largest real world data comes from Resumab with 550,000 injections. Obviously, they started first. So this I've already told you, Resumab has approval for all the indications. Renia had accept ROP. Ranizurel, as of now, labeled indication is neovascular AMD. Now I'll touch upon few of the cases just to show you sometimes there is a inhibition and people think that whether they would work the same. Just to show you that these molecules work as good as your innovator molecules. Resumab, you must have seen real-world prediction. Deb Dulal is sitting there. So he had done wonderful work as far as real-world data is concerned from Disha Eye Hospital. So I would share some of the Renia's data from my practice. There's a DME case after 1 injection, resolved well. Means I'll quickly go through this. Again, DME case, NSD with IRF resolves very well. Again, comes back because again 4 weekly injections. We shifted on a CM scheme, Tamil Nadu, where I practice. CM scheme, Avastin comes free of cost. Again, diabetic, responded well. RVO, again, NSD with IRF, responded well. These are all Renia's cases. Again, RVO responded well. Neovascular AMD, after a couple of injections, you see hemorrhagic PD goes off. OCT looks pretty. And again, you see a parapapillary CNVM that also responds very well. Means a spectrum of cases just to show that it is efficacious in all the indications. So do not have negative perception about these molecules. That's the way we can serve this country, and that's what happens in the last 7 years. Where we were in 2016, that's where United States and Europe stands as of now. I was in Portugal last month. I was in a debate that whether these molecules can rule US and Europe as well. I said, this would happen because we also started the same way with the negative perception. I was a big critic of biosimilars. But now means I can say with authority that I can use that in my near and dears. And Europe is testament. There's no molecule which has been withdrawn for safety reasons. Future lies in biobetters. And this is the last slide. What I would say is there is a possibility of waiving of phase 3 trials going forward. We are talking today. I would see you all after 10 years, and that might happen. Already UK has taken a step for ways, because that's how you can transfer the real benefit of these molecules for clinicians, because novel assets could be unpredictable. For example, brolucizumab. But biosimilars, they already have a track record and they emulate a molecule which has safety and efficacy established. So the chances of surprise are much, much less. And if we believe them, I'm sure we can change many lives, not only in this country, but across the globe. Thank you for your kind attention, and I'm happy to take any questions.
Thank you so much, Ashish. The entire world is moving towards biosimilars. And thank you for this loud and very vociferous talk on the utility, and what Anand said, that you see anti-VEGFs have potential to reduce the blindness by half. And here is the role of because you see any treatment launched by anybody should not only be effective and safe, but should be affordable also. And hence the role of biosimilars. And I'm sure Intas will carry him on their shoulders once they go out, the way he started. But one question to you is that in your experience, although Razumab has the largest, in terms of biosimilars in India, and was the first biosimilar to be launched all across the globe, any difference between 3 or 4 biosimilars which are available, whatever data we have about Reliance and Ociva and Razumab or Vijumab? I personally feel there's a tug of war now between the rate reduction and the schemes which company gives. So your take in very brief.
So as of now, sir, I think it is difficult to point out that because all the trial designs were similar, with loss of 15 letters. The only one thing which I would say what Lupin has done well means they are analyzing much deeper. What they have analyzed in the recent past is that
let's not are they all of them efficacious?
All of them are efficacious, because we have to believe on the trial.
All of them are safe?
All of them are safe.
So given the choice, whosoever gives me cheapest, I'll purchase that.
Cheapest, yes. Financial dynamics.
That answers my question.
Yes, absolutely.
Although the Razumab has the largest, still lakhs and lakhs of vials, where all these small babies are just coming up. That's it.
No, what I was trying to suggest is, for example, when Intas Razumab was launched, if you have noticed, there was a paper from Basel, Switzerland, by Novartis head office.
Yeah.
What they did was they did compare some of the batches of Novartis with some of the initial batches of Intas. So I would suggest, even Intas, Retina, whosoever is there, inter-comparison little bit should happen in terms of the more, deeper details of endotoxins or, structural similarity. Means that would give us some more clues.
So that is your job. That I think you should do that. So one question.
Thank you so much.
Thank you. One thing I would like to share is when the first Razumab came in, and in fact, Dr. Anand will remember it, we presented that paper in 2016 in ASRS in San Francisco.
They will, you can be ready.
And then I was surrounded by the media people, the press, the medical media, that because when I was studying in Toronto, there was $1,600 to $1,700 each Lucentis. And they were surprised that how could you make an anti-VEGF of around $250. So that was the challenge which they took it up, and then they argued with the government how can they bring down the rate of the Lucentis, because it was costing very heavy for the Canadian or the American government. So they tried their best to do that, but we are now happy that we are getting a lot of molecules which are pretty safe and pretty efficacious. But one thing I would like to say is when Dr. Ashish says unpredictability of the brolucizumab, I think each of these anti-VEGFs in this thing, they have certain of teething problem
No,
or
in any
hiccups
case,
which
we are
are
not we are
faced.
not discussing about
Yeah,
brolucizumab.
so
Our
he put
topic
one
was
slide there. But I think the time has to go really to come out of these kind of inflammatory reactions.
You
And
see, that's perfectly okay. You see, my intention here was biosimilars have entered. All the innovator molecules have a patent life, and these biosimilars, irrespective, they are safe, they are efficacious, and there should be no hesitancy in using them.
true.
That's the message to be conveyed. And with this, I will request Debulal to tell us. You see, from the biosimilars, we now move on to newer anti-VEGFs. And believe me, all newer anti-VEGFs, they will have a, some patent life. After that, again, biosimilars start coming. And even you see, happy news is that although India and Intas led the way, but US also recently, as he showed, has approved biosimilars for their practice. Yes, Devula.
So thank you, sir, for this kind opportunity. And I'll be speaking on newer anti-VEGF. And so one thing that we've gone through in the past presentations is that anti-VEGFs are the way forward, and they have made a transformational change as far as retinal vascular disease goes. And today we talk about improving vision rather than preventing blindness. But the problem doesn't end there. There are economic issues, there are agents that do not work on some patients, and of course we need durable agents which can actually work for a long time. So anti-VEGFs that we have today are good, but they are not absolutely perfect. So this is what I'll be basically touching upon. The ones in green actually are available in the market, in at least in some part of the world, if not in India. And the ones in yellow are in various advanced phases of trial, and I'll just touch upon them. So basically, we'll be talking about safety and efficacy of the new agents. And to start with, the port delivery system with ranibizumab. So this is something that addresses the issue of durability. And this is basically a refillable intraocular implant, and then you pump in ranibizumab. You need a small procedure, surgical procedure, to put the implant into the eye. Has been approved by FDA, but there have been some issues with it. So what it does, it releases a little bit of ranibizumab each moment into the eye. And if you look at the results, then the visual acuity performance and the anatomical performance is good if you compare it with monthly ranibizumab injections. But the main problem lies in the fact that ocular adverse events of special interests were pretty high. You had 1.6% implant dislocation, you had rhabdomatogenous detachments in close to 1%, and 1.6% endophthalmitis. So you have a more than 2 to 3% adverse event issue. So current status, it has been withdrawn from clinical use, maybe temporarily, we don't really know. So next is brolucizumab. So many of us have used brolucizumab for some time now. So basically this is a drug that's kind of non-inferior to aflibercept as far as the Hawk and Harrier trials, sir mentioned red carpet trials. And if you talk about the fluid resolution, then to quote sir only, that it is a drug that has greater power of fluid resolution. Having said that, in the kite and kestrel trials also we see a similar story, similar kind of visual acuity improvement and little better fluid resolution. But the issue with brolucizumab is, we are scared of, something called adverse events that have been noted. Initially, it was 4.6%. With kite and kestrel, it is 1% to 2% with brolucizumab versus aflibercept, and 2% and 1.7% in the kite study. So talking about a larger data, any form of IOI or RO was noted in this Iris Komodo healthcare map at around pegged around 2.4%. But we've also had some Indian publications with brolucizumab, including our publications, and what we've noted is that it is lesser than that reported in the West, and we've been able to treat these patients well with topical or some oral steroids. Moving on to the next drug, faricimab. It is probably about to be launched in the country, but just to bring in some context, so we have, other than VEGF, we have certain other things which are also responsible for vascular stability, and angiopoietin 1 and angiopoietin 2 are essential for vascular stability. Normal situation, you have angiopoietin 1 binding up with the tyrosine kinase, transmembrane, molecules, and they actually maintain stability, whereas in diseased condition angiopoietin 1 is competitively inhibited by angiopoietin 2, and that's where faricimab comes in. Now it is a bispecific antibody which has a FAB fragment which is anti-angiopoietin 2. It has a FAB fragment that's anti-VEGF-A, and you have a modified FC which reduces systemic exposure. So you have a drug which is supposed to take care of everything. So if you look at the trials for DME, the Yosemite and the Rhine, the visual acuity performance is pretty similar to aflibercept. And if you look at the anatomical outcomes, they are also pretty much similar. So if you talk about durability with faricimab, this is where some difference comes in. Close to 80% of these patients could be treated with faricimab once every 16 weeks post the loading dose, of course, and post 12 weeks I should say 80% of them could be treated. And regarding 16 weeks, Q16 weeks, you have around 64% or 60% here in the Yosemite that could be treated every 16 weeks. So this is something that's nice and important. And if you talk about adverse events, then you have a figure of close to 3% with the Yosemite Rhine, 3% with faricimab, as opposed to close to 2% with aflibercept. So time will tell whether this is there in the real world or not. Talking about NAMD with faricimab, the Tenaya and the Lucerne have a similar story. There's improvement of vision that was noted, and there's a significant reduction in the fluid compartments as well. So moving on, again, little bit of concern maybe, but real world data is yet to come. 2.6% for faricimab versus 1.52% for aflibercept, the serious adverse events that were noted. Again, you have a situation where you have a lot of people who almost close to 45% who could be on Q16-week dosing for neovascular AMD, and you have patients up to 80% who could be sustained on 12-weekly dosing. So moving on, some of the other things that are there. So to enhance durability, you have something called KH301 in phase 3 trials. It's an antibody biopolymer conjugate which enables a higher dose for longer treatment duration. So basically you have ranibizumab with some polymer that can last for a longer period of time. Next is OPT302. What about the recalcitrant patients who don't really respond to VEGF, anti-VEGF molecules? So it is suggested that VEGF C and D go on an overdrive when you treat patients with anti-VEGF molecules. And this is where OPT302 comes in. It attacks VEGF C and D, and thereby you have better results. And what they have shown is that 5.7 letter gains in these lesions, and reduced fluid and greater reduction in total lesion area, CNVM size. That is what they have been telling. So we are hopeful that it comes across. Then talking about alternative pathways, we have something called plasma kallikrein, which is being used for DME, and this THR149 is a selective plasma kallikrein inhibitor having potential to treat 40 to 50% of patients who respond suboptimally to anti-VEGF. Next, you have something called THR. This is of course THR149. Next, you have something called UBX1325. So the root cause of this VEGF liberation is senescent cells. So if you can do away with the senescent cells with something like UBX1325, then you have a situation where you don't need much of anti-VEGF. And the last part of the presentation will have a little bit about genes. These are vectors that are used to deliver genomes that induce production of anti-VEGF within the eye. So with RGX314, this is a subretinal injection, little cumbersome procedure, but it works all right. And what effectively it does is that it reduces the necessity of repeated injections, to put it in a nutshell. The next one is even better. Similar thing, where you have a promoter with which ultimately will help the retinal cells produce aflibercept, and it is an intravitreal injection, more in our like everybody's domain. So next is how does it help? Again, similarly, the necessity of intravitreal injections is lessened, and you have also multi-mechanistic gene therapy models where different types of anti-VEGFs can be produced similarly within the eye. And again, it's important that these are being tried out in a intravitreal injection format. So there's hope at the end of the tunnel, and I'll take this opportunity to welcome you all to the next AIOC. Thank you, sir.
Yeah, excellent overview, Dr. Devdudal. Actually, I think ILIA or aflibercept was definitely a major jump over the pre-existing ranibizumab, which was a jump over bevacizumab. And now we are really looking forward to faricimab because the durability criteria which you said, there is a almost 20% increase in the number of patients who can be extended to Q16, and similarly another 20% who can be extended to Q12. So definitely it's helping, and also the anatomic and visual criteria are superior to the aflibercept in all the studies that have been done on AMD, DME, retinal vein occlusions. So that being the case, we do look forward to faricimab.
Wait till October. I think October is the month.
The trial has already started. Dr. Debulal, have you started using faricimab trial, or are you inducted?
No, not yet.
I think Chennai it has started. Yeah. Yes, Ashish, you wanted to say?
So I would just like to highlight, means whenever we present faricimab data, we talk about Q16, Q12. But if you really see, the trial was not designed to prove durability here. The answer was not durability. But the reason is, if you look at the aflibercept arm was not allowed for Q16.
Yeah.
Yeah, it was not a comparative study.
It was not. And if you look at the outcome measures were not to prove durability. It was just to check how efficacious the drug is and how safe the drug is. So even the trial results, if you see, they clearly say it was not, durability was not an outcome. So sometimes that notion goes in a wrong direction when you do not have apple to apple comparison.
So see, you are right, because a lot of aflibercept patients, in case that trial had allowed them for Q16, maybe.
But drying was definitely better. That's what we saw.
Drying, in any case, brolucizumab also was better.
Even the drying was not statistically significantly better. It had trend towards being better.
Trend towards better.
And I agree with you that it was not compared. The duration was not compared between the two drugs. So I agree with you. It may not really.
So we just need to think a little deeper.
Correct. Exactly.
Thank you. I think we need to move on, because this, I think, should be the most important talk from our perspective, that is pharmacoeconomics, and none else than Dr. Pradeep Kumar, who has done a lot of work on economic aspect of usage of antivirals, especially pertaining to this part of the world. Pradeep, are you there? Yeah.
Very good morning, sir. The presentation can be changed. It's Dr. Thirumalesh's presentation here. So I'll be talking to you about pharmacoeconomics. Presentation change.
एक presentation ऊपर वाले के ले लेना.
Dr. Pradeep.
Dr. Pradeep करके. Pradeep Kumar.
Change करना है.
Yeah, now we have the
this is okay.
Presentation. Thank you. So I'll be talking to you about pharmacoeconomics of anti-VEGF. I'm Colonel Pradeep from Army Hospital Ara. We have no financial interest in whatever we are going to say. The data has been obtained, thankfully, from the OEM sales reps, and the union government data is from .gov websites. I am thankful to the corporates for sharing the data of their sales. And there are three ways of looking at pharmacoeconomics. One, from the point of view of the doctor. Second, from the point of view of the patient and the government. So I'm going to try and amalgamate these 3 point of views. And what I want to share with you is help everyone understand pharmacoeconomics, to show you how the government is planning to intervene in your clinical practice in future, how you can empower by presenting evidence to Health Technology Assessment Initiative of India, and how you are supposed to generate this kind of evidence. So first, to understand pharmacoeconomics, it is simply the science of telling us that when we compare the value of one pharmaceutical drug to another, which one is better. It basically dwells on the cost and effect of a pharmaceutical product. And the methods it uses is cost effectiveness analysis, where cost is measured in monetary units and the effects are measured wherever we find a lower ratio of cost to effectiveness, that particular intervention is preferred. So you have drug A, drug B, you measure the consequences and the costs, and you derive an incremental cost effectiveness ratio by the difference of the new molecule compared to the standard treatment and the benefits, and the one with a lower ratio is preferred. Next is cost utility analysis. So how is it different from effectiveness? Utility talks about the impact on the length of life and subjective levels of well-being. So quality associated life years is the best known utility measurement scale, and one QALY represents 1 year of perfect health. And when we talk about utility, this particular word utility we'll all have to remember in next 4 to 5 years to come. It talks about perfect health at ratio 1 and death as 0 and different levels of utility for various diseases. Next is cost benefit analysis, where we turn the benefits which are accrued from a certain intervention into monetary value. So the monetary value of vision reduction has to be assessed before arriving at a cost benefit analysis. And the costs which are going to be assessed are not only the direct costs, but the indirect costs of treatment and the indirect costs of the impact that is going to have on the patient's life. And where the government comes in, it is the cost minimization analysis. This is where the government tries to figure out all the three methodologies and tell us, the doctors, that you're not supposed to use branded drugs but generic drugs. So what is the government doing as of date? Let's talk about direct benefits. Our union budget has increased from 84,000 crore to 89,000 crore over the past 2 years. But the budget allocation for NPCB comes under tertiary care program. And the tertiary care program has suffered because of the impact of COVID from 405 crores to 288 crores. When we talk about the blindness part, it is just 1/10 of this entire tertiary care program. Effectively, it gives you 3 crore rupees for retinal health if we talk about it. So where is the government reimbursing all the retinal health procedures that are going on today? By supporting the insurance agencies, that is CGHS, ECHS and Ayushman Bharat. I'll talk about it later. And giving institutional support to various central and state government hospitals. Retinal disorders, as has been agreed in all these previous talks, is a leading cause of visual impairment. And the problem occurs that the functional effect of loss of vision becomes extremely important in adulthood and older life when diabetic retinopathy and AMD become the key players in reducing vision. And the debilitating nature of vision loss includes 40%, you remember this figure, 40% in direct medical cost and close to about 60% divided equally between the direct non-medical costs which have been shown here and the indirect costs of productivity loss of the patient. Let me talk about one particular disease. The disease burden of AMD in India. If you look at this slide, we can calculate that there is a requirement of approximately 6 lakh patients requiring anti-VEGF injections per year. And these are the sales figures which have been shared with us. When we talk about the innovator molecules, there were 1,14,364 sales in 2022. We talk about Razumab, they claim they have sold about 1,70,000 such units. And Ozurdex just came to about 33,600 units. Now, if we try to break down the unmet need, the requirement was of 18 lakh injections, while the approved drugs were only 3,18,000 sold. So where are the rest of the 15 lakh injections coming from? Either they are not being given, so the patients are going without treatment, or that gap is being fulfilled by bevacizumab. The cost of approved injection alone is 540 crore, and that too only for one disease, which is AMD. The government allocation cannot be beyond 540 crores by any means. So there is a huge difference in the budget allocation and the treatment requirement. Government is claiming that they are reducing the out-of-pocket health expenditure. They are producing such graphs where they are telling us that your out-of-health expenditure is now reducing from 64% to 45%. But please understand that any expenditure in a country, whether reimbursed or not, is borne by the country as such. And whenever you include health insurance in the entire gamut of things, it is an overhead expenditure. It actually reduces the direct benefit which is given to the patient from the government. What is this health technology analysis? So health technology analysis is an evidence-based decision making for healthcare benefit. It talks about two very important things in today's discussion of anti-VEGF. It talks about ethical issues, whether to use an approved drug and an unapproved drug also for giving safe and effective health. Now, the applications mainly require the decision making to tell us that what will be the reimbursement procedures, what will be the SOPs where doctors will have to follow, and the pricing and procurement of drug. This is where the corporates come in. The government will be empowered to talk to the corporates to reduce the price of certain drug if they want to practice in our country. Now, how we have postured ourselves. In 2014, World Health Assembly told us to put it in place. By 2018, the HTA in India was established. It is maximizing health by reducing out-of-pocket expenditure and, most importantly, reduce inequality. Both the rich and poor should get the same treatment. That is the objective. Now, this is the structure of HTA. Now we see that they are the ones who are going to be directing the policies. But who are going to give them the evidence? They have said resource hub, technical partner, and stakeholders. Now this very important part is stakeholders. When have we seen an eye specialist walking up to the government and telling them that please change your policies? This is where you and I have to come in and play our part. The current evidence of pharmacoeconomics has been amply explained in today's talk. We know that there is blindness. The blindness has not only direct and indirect costs. The health-related quality of life of blindness by AMD is almost comparable to be that of stroke. This is where and how we have to talk to the government, that if you're allocating budget for stroke treatment and coronary artery disease treatment, it has to be similar to the treatment of AMD also. This part has been amply covered, so I'll straight away jump to the direct cost 40%. Please remember this figure, and indirect cost 60%. So in real life, we see that pharmacoeconomic intervention can help healthcare professionals to make an informed and efficient decision. Within finite resources, we can distribute the benefits of these kind of anti-VEGF treatment to all. Now, the source of funding in India are not going to change. Between 2021 and 2025, they are going to remain equal. They can only be distributed into various subheads. So what and how we are going to deal with it? They were the crux of the equation comes. Let me talk about two molecules, aflibercept and bevacizumab. Here, in place of bevacizumab, I can replace it with ranibizumab, razumab, rani, any of the molecules which are 4-weekly acting. If I talk about aflibercept, the per dose 50,000 rupee approximate expenditure, 3,000 rupee expenditure in case of bevacizumab. But the follow-up with aflibercept is slightly lesser compared to what it is required with bevacizumab because the drug is more durable. So you see only 60,000 rupee expenditure in the indirect cost, where a 1,20,000 rupee expenditure in indirect cost with bevacizumab. Still, the drugs are not comparable. The aflibercept part will require 3 lakh rupee per year, while the bevacizumab arm will require 1,50,000. That is twice the expenditure. So you cannot explain to the government that please go ahead with a 3 lakh rupee drug compared to 1,50,000 rupee drug. How this can change? Just a small change. You move from aflibercept to brolucizumab. Here you can replace brolucizumab with faricimab or any other longer acting drug. And here you see that the number of visits of the patients are going to reduce. And now we are talking about comparable economic impact on both aspects. So these are just two examples how pharmacoeconomics can be used to advise the government. But let me come back to this particular example. It does not mean that aflibercept does not have a role. What the government is supposed to do is talk to the corporate and help reduce the cost of the drug across the country, which is achievable, and it can be achieved. Sorry, these were few cost calculators which I was supposed to cover. These are another method of pharmacoeconomics from patient's point of view, where a patient can also calculate the impact of the cost that is going to be consumed during his or her treatment. But these calculators are not the solution. The solution is to achieve cost reduction by using longer acting and more durable molecules. And we have to suggest to the government to reduce the pricing. The pricing is by reducing the tax. And how we are going to convince the government in doing so is by showing this kind of evidence. So I'll request that us, AIOS as a body, should now develop a health technology assessment bureau inside our organization, which has the authority to advise the government. Because if this task is left to certain government institution, I also belong to a government institution, it is not going to be enough because the voices are going to be lower. When all of us combine together and we have our unified voice going to the government and advising them that this is what you're supposed to do, they'll listen to us. Thank you so much.
I wish we had some bureaucrats or politicians sitting here to listen to this talk because most of us understand this indirect costs and the need to urge the government to reduce this cost. Avrinder, you have any comment or observation?
One thing,
Mike.
one thing I'd like to comment is that there should be a AIOS committee and people from government. And like what we have in American Academy, there is a session how to influence the insurer and the government agency. So we should come and there should be a central negotiating body, like insurance people are negotiating with an individual practitioners or the group, and they are giving different rates. So AIOS and the government should come together like ECHS and CGHS to negotiate a set protocol so that everyone in the society benefits from it.
Wonderful, wonderful idea. I'll pass it on to Dr. Harbans Lal. That's what I can do. Thanks. Maybe I'll, borrow this talk from you because
Thank you so much.
I don't want to reinvent the wheel, and because the depth which you have got, I also have delivered one talk on economics, but yours is much more better than what I had done.
Kind of.
Yes, sir. Tirumalesh is ready. Yeah, please.
Yeah, Dr. Pradeep, very fantastic talk. So you have done a pharmacoeconomics between bevacizumab with aflibercept and with the brolucizumab. What about the difference in case of Ozurdex usage in case of DME? Because that's the maximum cases where we find the diabetic macular edema or the diabetic retinopathy is more common in India. And does the pharmacoeconomics change in this, or what you made was specifically made for CNVMs and the PCV, or based on the drug duration and the availability?
The talk was specifically made for anti-VEGF, so that's why I only presented one slide of Ozurdex. But if you look at the intervention that UK have advised, they followed NICE. NICE is their HTA body, which advises the government to tell them that in first onset of diabetes mellitus, diabetic macular edema coming to you, you are forced to give Ozurdex. So that is where the difference comes in. And yes, they are able to achieve a cost reduction by using intravitreal steroids as the primary treatment in cases of diabetic macular edema and macular edema associated with CRVO. And this happened because of this HTA body called NICE in UK. So this is where I am suggesting that before such an intervention happens to us in India, where we are not able to control it, we have to raise our voice together.
Okay. Thanks, Pradeep.
Thank you, Dr. Pradeep.
We invite Dr. Tirumalesh, because the reason of asking Tirumalesh to participate in this program was so much of burden, so much can we titrate and prognosticate based on biomarkers. Yes, Tirumalesh.
Thank you, sir. At the outset, I would like to thank Dr. Lalit and also the scientific committee for giving me this opportunity. So the topic that was given to me was biomarkers and the road ahead. Can we really titrate or prognosticate? Now, what is the understanding till now is what is a biomarker? It is a measurable indicator of a biological process, or it is something that helps us to determine whether there is a response to a therapeutic intervention or not. It may be a biochemical biomarker or a molecular biomarker. It can be a genetic biomarker which can tell whether a disease is going to be more severe than others, or imaging-based biomarkers. We as ophthalmologists tend to use more of imaging-based biomarkers, and every single time we tend to do the treatment based on it because it is repeatable and available to us. This can be used to diagnose, monitor, and also predict the disease progression. It can help to predict the response to treatment as well. Now, biomarkers enables us also to understand the underlying biological mechanisms of the disease and to identify potential therapeutic targets. So if you're using only one type of biomarker, you might not be able to understand what is happening at the microcosm level. So looking at only a single type of biomarker might not help us to look at the full picture. So any ideal biomarker should help us to make more accurate diagnosis, understand the disease process, and also herald the way for more and more customized treatment, because we all know that the same injection can produce different effects despite having the similar kind of an OCT. Now, what is known to all of us is this. The commonly used ones are the OCT biomarkers. You know that if you have a hard exudate at the fovea, it means that the patient probably will not improve in his visual outcomes. It is also a marker of chronicity. Usually it takes about 6 to 12 weeks for development of a hard exudate once the edema disappears. Also, sudden disappearance of edema can suddenly precipitate a hard exudate. If it happens at the fovea, it is a bad marker. Neurosensory detachment, if it is there, we look into the systemic associations. Is there any presence of hypertension, hypoproteinemia, etc., etc.? And that again, based on the image, can tell you about subsequent disease. There are others which are very clearly seen on the OCT, which are like bridging processes, which will tell you that, oh, they might respond to treatment. VMT typically changes the way we manage from medical management to surgical management. There are a few others like pearl necklace sign and large cystoid spaces, which typically indicate that the disease has been more chronic in DME. Now, similar biomarkers can also be seen on the OCT for AMD. So we tend to classify the disease as type 1, type 2, depending upon where the membrane is, and subretinal fluid definitely means that there is an activity, which means that you continue with your treatment. Pigment epithelial detachment will tell you that the vision does not get affected, but this will probably be a patient who will be taking a very long treatment. Something like a precordial cleft can tell you that, yeah, the disease is active, but it's not going to rob the patient of the vision. Outer retinal tubulations mean the patient is at the end of the disease, and hyperreflectivity dot means that these patients are going to respond relatively less than what is given. But what I was called for, or when Dr. Lalit called me for this talk, was for this. Now, we are looking at one part of the disease. What happens if we try to understand the disease to look at the molecular biomarkers? So if you look at aqueous humor of patients who are taking treatment, you can see that it reflects the true nature of the disease. We treat the patient by giving anti-VEGF usually in most patients of diabetic macular edema. So naturally VEGF-A is high, but what happens over a period of time is, if you look at a patient who's been recurrent, the VEGF levels are not going as up as when it was treatment naive. So there are other factors which are there, like the soluble LICAM levels, which tend to become more and more up, and the inflammatory factors like IL-6 also tend to go up. There are extracellular modulation factors like MMP9s, which tend to go up. Integrin 1 beta and IL-1 beta also is a chronic inflammatory marker. So whenever there is a chronic inflammatory marker up, there is also presence of IL-6 which is going up, and soluble LICAM levels going up. So this is one thing that can tell you that the disease is entered into a chronic inflammatory phase, which is as much influencing the disease as VEGF-A. Now the common argument is, okay, we are giving VEGF, it is trying to take it's trying to give you good enough vision or keeping the patient under good treatment. So why do we need to see this? This particular thing will help us in more and more tailor-made treatment, but of course we need more and more targets for that. Currently the target that is available is intravitreal steroid, but what steroid does is it kind of blanketly suppresses everything, both the good and bad part of it. And another problem with steroid is it can cause glaucoma and cataract. That is what keeps us from using steroid as the primary line. So if individual of these can be blocked particularly, that can help in treating the patients better more and more in the future. So this slide actually indicates that these biomarkers can be looked into if we are trying to customize and tailor make the treatment, but the availability of targets is something that we look forward to. These are the ROC curves. Now, the other question that many people ask is, okay, if I look at VEGF levels, can I tell the disease is absolutely active? Now, you can see the distribution here. Not every patient of diabetic macular edema has a very high level of VEGF. There can be other things which are kind of going up and down, like I showed in the previous slide. So you can see the clustering that is there, the controls and the DME, they are relatively there, but there are few patients where they're going up. So based on our knowledge of understanding of pathomechanism in printed literature, we tend to say, okay, whenever there is DME, there has to be an increased VEGF level, but that's not really the case. If you put an area under the curve and you look at what is the sensitivity of it, the sensitivity is only 71.43%. Now, if you look at ICAM, now ICAM actually correlates more than VEGF-A, but the commonly used drug that we are doing in current practice is VEGF, which means that we are not exactly treating the pathway or the way in which the disease should be treated, or probably there are new targets that are upcoming. Now I'll show you an example case where we had taken aqueous humor and correlated with the OCT. This thing, you look at the level of cystoid space probably is inflammatory, or but you can see that the VEGF level is very high. In another case which had cystoid spaces, but there the VEGF level is too low. But both the cases responded to anti-VEGF, which means that anti-VEGF probably has a polymorphic effect. It might be affecting other pathways as well, but then over a period of time it tends to lose its effect on those receptors because they tend to get saturated. Now this is another patient where you can see that there has been a macular edema where I gave an injection. So the first one is when the patient presented to me, despite having about 1000 microns in edema on the OCT. In OCT, you can see that after I gave the patient an injection of anti-VEGF plus steroid, the level of VEGF actually fell down even further than what would have usually happened, right? But after about 2 months, the patient's VEGF level came back. What this example indicates is that tailor making of treatment is possible. If you tend to target individual biomarkers, probably it can lead to lesser number of injections rather than just looking at cheaper alternatives. If we understand the disease, probably we might treat it better. Now, another question that is commonly asked to us is, you have to take an AC tap, so this is an invasive procedure. So what we are working at Narayan Nethralaya is this, that probably look at all the biomarkers, keep the data, analyze the data, and probably correlate it with tears. Although we are not there yet, we have analyzed about 40 factors, out of which only 3 to 4 kind of correlate between tear and aqueous. So a day might come when somebody can take tears and put it on a multiplex ELISA like that, read it in a machine and give you a printout where you can understand the pathways that are involved and what is the current stage of the disease, and probably choose your anti-VEGF injection. Thank you so much.
One remark I'd like to make. Most of these biomarkers are really under study, evaluation, and should not really guide our clinical judgment. Like to say that if you see hyperreflective dots, just go to steroid immediately, or hard exudates, go to steroid, subfoveal fluid in DME, go to steroid, is wrong. Because steroid has too many side effects to take it lightly, and the anti-VEGFs work as well. There is no studies to conclusively show that steroids or anything is better than the other with these biomarkers. With the exception, I would say, of RP detachment in AMD PCV, where definitely aflibercept or the stronger molecules like brolucizumab may have an advantage over ranibizumab. Apart from that, I don't think any biomarker should make a decision for us.
Yeah, biomarker is basically to understand the disease and probably know where you are at. Like you said, NSD probably also look into systemic issues rather than just say go for steroids, because giving a steroid can suddenly cause precipitation of hard exudate at the fovea, which we really don't want. But that can happen with anti-VEGF as well.
So, yeah, I mean,
because we have run out of time, we should respect the time. Thank you all the speakers, and I think we have had very useful presentations by all the authors. And I thank Mallika, Srinivas, as well as Vrinder for helping me conduct this national symposium. Thank you very much.
Thank you, Lalit sir, for conducting it so well. Thank you.